Valentín Ceña is Professor of Pharmacology and Medicine at the University of Castilla-La Mancha, Albacete, and he's the head of the unit Neurodegenerative, and his talk today is entitled Nanomedicine, the Future for Therapeutics of Neurodegenerative Diseases.
First of all, I would like to thank the organizers for inviting me to present the data today. During the next few minutes, I will try to convince you that nanomedicine has a place in the possible therapeutics for neurodegenerative diseases. Basically, first question is, what is nanomedicine? Nanomedicine can be defined as the use of nanoparticles as a part of a diagnostic or therapeutic process. The next question that comes is, what is a nanoparticle? And a nanoparticle is just nanoparticles are multifunctional materials that interact with biological systems in a controlled way. They exhibit unique properties due to its small size, and one of these properties is that the percentage of atoms that are in the surface of the molecule, and so they can interact with the environment, is very high. In terms of size, nanoparticles are really in the range from 1 to 100 nanometers. That's the name of the nanoparticles. And they are compound for many different chemical types. You have dendrimers, cationic lipids, polyethylenimine nanoparticles, carbon nanotubes, chitosan nanoparticles are representative of natural polymers, collagen nanoparticles, quantum dots, carbon nanohorns, gold nanoparticles, and iron oxide nanoparticles. There are some more, but let's say that these are the more important. We have been working with dendrimers, carbon nanohorns, and carbon nanotubes. So all the data I'm going to present here today has been done using dendrimers, different types of dendrimers. This is the question: are nanoparticles potentially useful for the treatment of CNS diseases? So I'm going to present one example of the two possibilities that we have. One possibility is the use of nanoparticles as therapeutic molecules by themselves, without any addition. And the other is the use of nanoparticles as carriers for therapeutic molecules. And the therapeutic molecules can be either a drug or genetic material. Let's start with nanoparticles as therapeutic molecules. And we have some dendrimers that are anti-inflammatory by themselves and can be used in the treatment of multiple sclerosis. I'm going to present data only on in vitro results, because we are just starting the experiments with whole animals, and we don't have yet conclusive results. Everybody knows here that multiple sclerosis is an autoimmune disease that affects a very high number of people, 2.3 million, has a high social and economic impact, is chronic, it comes into bursts, and reduces life expectancy by 10 to 12 years. The main initial molecular fact in multiple sclerosis, basically inflammation, that leads to demyelinating at a later stage, a section of the axons. So inflammation is the primary cause through an autoimmune attack of multiple sclerosis, and so anti-inflammatory nanoparticles like phosphodendrimers might be useful to prevent the disease progression. So we have studied the anti-inflammatory properties initially in human monocytes and macrophages. And the first thing is these phosphodendrimers, you can see here, are not toxic, even at 5 or 10 micromolar. This is a very simple scheme of what happens during innate immunity. NF-kappa B is translocated to the nucleus, induces the expression of inducible nitric oxide synthase. Nitric oxide is secreted, and inflammation, vasodilation, and VEGF-induced adhesion molecules are expressed. Phosphodendrimers inhibit all the chain of facts on this signaling pathway. Initially, they inhibit NF-kappa B translocation to the nucleus. You can see here that LPS is used to induce the translocation and induce inflammation, and there is a clear mark effect of both dendrimers that I'm presenting here, and this is the MG132 that is the standard control for this type of experiments. In second place, phosphotendrimer also reduce LPS-induced años expression, as will be expected, due to the inhibition of translocation on FB kappa B. You can see here it's a partial reduction, and also reduce CDA disease expression, indicating that macrophage monocytes are not really ready to be activated. We have repeated this data using dendritic cells, and we got basically the same results. Also, these phosphodendrimers inhibit another fact that is very critical in multiple sclerosis, is this lymphocyte proliferation. You know, lymphocyte proliferation can be induced by a combination of stimulatory antibodies against CD3 and CD28. You can see here a mark. These are also human lymphocytes. You can see here a mark-induced induction of proliferation by the combination of the antibodies, and for this dendrimer, there is a clear dose-response inhibition of the proliferation. So, in conclusions, we could say in this part of the talk that phosphodendrimers are not toxic to macrophages. They present anti-inflammatory properties. Phosphodendrimers inhibit inflammation at different levels, and if kappa B translocation to the nucleus, años expression, and nitric oxide production, and also that phosphodendrimers inhibit lymphocyte proliferation. These molecules are potentially useful to be used as scaffolds for developing drugs useful to treat diseases like multiple sclerosis, where inflammation plays a key role. I would like to result scaffolds, because this is just a plain dendrimer molecule. And on top of that, as we will see later in the talk, we can incorporate different signaling or imaging groups. The second example I want to present is the use of nanoparticles as carriers for therapeutic molecules. This is an example of a nanoparticle. In this case, it's a dendrimer, but you can think on any type of nanoparticles. And the advantage of having so many functional groups on the surface is that you can append to this molecule, by chemical modification, different groups. One targeting group that is here represented by antibody, just to direct it to certain areas or cells. Localization group to have an image probe, or therapeutic groups that can be either a drug or siRNA. The data I'm going to present is just using siRNA. So the first question is, are these nanoparticles able to transport therapeutic molecules into the CNS cells? And for that, we use to study a simple biological problem. I'm trying to answer this question. What is the role of coffulin in a cytotoxic neuronal death? You know, actin binding proteins like coffulin regulates actin filament depolymerization. In the functions in which the actin microfilaments are involved include mobility, cell division, morphogenesis, and protein traffic. Cytotoxic is mediated by activation of inotropic glutamate receptors that increase intracellular calcium, and that triggers an intracellular response that includes translocation of certain apoptotic proteins or proapoptotic proteins like BEAT or BCLX to the mitochondria, the release of cytochrome C, and the beginning of the activation of the effector caspases. So when NMDA induces VAS translocation to the mitochondria, following an increase in intracellular calcium, you can see here, this is VAX in the mitochondria. At 3 hours, there is an increase in VAX to mitochondria, and this causes mitochondrial depolarization and also neuronal death. So this is a very classical phenomenon. On the other hand, we know that coffulin is activated by dephosphorylation, but phosphatases from the slingshot family of phosphatases, and is inactivation by phosphorylation by the link kinases. But coffulin is also activated during cytotoxicity by the phosphorylation with a very similar time course to VAX translocation to mitochondria. You can see here that at 3 hours, there is a marked decrease in the ratio of phosphorylated coffulin to to coffee. In other words, is activated at 3 hours. You can see here, just to refresh your memory, that at 3 hours there is also an increase in bus translocation to the mitochondria. So the next step was rationally to perform coffee lack of function studies. But the problem in this case is coffee is knockout mice is embryonic lethal, which limits a lot the possibility of the studies. So we decided to use sRNA and use a dendrimer to vehiculize the sRNA. Generally, the basis is, this is general principle of biology. Most of the small drugs tackling some diseases interact with proteins that are either misfunction or overexpressed. But we decided to use sRNA to block the specific RNA coding for this protein. sRNA is very specific, degrades selectively the target mRNA, is not toxic but requires delivery to the cell. So that's why we're using nanoparticles. This is the effect of the dendriplex, the complex between this dendrimer, this nanoparticle, and sRNA on coffee levels. You can see here, this is the control levels. The nanoparticle doesn't do anything. The nanoparticle plus scramble sRNA that doesn't code for anything does nothing. But you see, when a specific sRNA for coffee is transported inside the cell, about only 15% of the original levels of the proteins remain in the cell. So it's very specific to decrease the levels of coffee. So what will be the effect of this decrease in levels of coffee on cytotoxic neuronal death? Basically, you can see here that in this case, bus translocation to mitochondria is markedly reduced. There is a partial protection for the decrease in mitochondrial potential, for mitochondrial depolarization, and there is a significant decrease in neuronal cell, just only knocking down in a very important level the coffee levels. So coffee removal decreases NMDA-mediated bus translocation, mitochondrial depolarization, and neuronal death. This is an example of using sRNA. What will be the advantage of the combination of a dendrimer and sRNA over a traditional drug, a small drug? Well, basically, the first one is better selectivity. You know, selectivity of the small drugs is just a matter of concentration. But in any case, with this case, you can get better selectivity providing that you go to the point number 3. The second is less number of side effects. But the main advantage is that you can prepare the nanoparticle to prepare cell type targeting. This cell type targeting, actually, we are able to direct a nanoparticle, not the same nanoparticle, but different nanoparticles, either to neurons, astrocytes, and some tumoral cell lines. This is achieved by just modifying the molecule. Some of them is just basically the basic chemical characteristics of the molecule, but you always can incorporate some targeting peptides or ligands to direct the nanoparticle to one or other cell type. The other question is, we are talking here about neurodegenerative diseases. So I have presented only in vitro experiments, but nanoparticles will go to the central nervous systems to have an effect, no? So, and the first point and critical point is, do nanoparticles cross the blood-brain barrier? For that, this is a cartoon of the blood-brain barrier, but for that, remember this scheme, the multifunctionality of a nanoparticle. We decided to incorporate an image probe to a nanoparticle. It was a dendrimer. And in this case, we incorporated a gold atom to the nanoparticle. Why? Because with this approach, we could visualize it by electron microscopy. So we injected the nanoparticle containing the gold atom into the tail of a mouse, and at different times, we fixed the brain and studied if we were able to find the nanoparticles in the brain. Here is in the cortex. You can see here the external pyramidal layer, the polymorphic layer, the external pyramidal layer, the internal pyramidal layer, and the polymorphic layer. You can see here, where the arrows heads are, there is a signal dense to electron microscopy, indicating that the nanoparticles that we injected in the tail of the animal reached the brain. Obviously, this is a first step, but really, this is the same in the cerebellum. We were able to identify the nanoparticles in different parts of the cerebellum. So some nanoparticles cross the blood-brain barrier, but another important point, for those nanoparticles that initially do not cross the blood-brain barrier, you can modify them incorporating peptides or signaling molecules to help them to increase BBB crossing. Now, the next question was, does the combination nanoparticle-sRNA have an effect on the brain? In this case, we use just the nanoparticle with sRNA, directed against one target protein that we use routinely, that is p42 MAP kinase. So in this case, here, we injected the nanoparticle in the tile containing the sRNA in the tile of the mouse. And you can see here, this is mRNA levels, that only the combination of the nanoparticle with the specific p42 sRNA is able to inhibit markedly in brain cortex the levels of mRNA for p42. But also, the same obviously happens when you use Western blot. This is the protein levels, the same effect. So, as conclusion, sRNA could be vehiculated to the brain by some nanoparticles to inhibit selectively a target protein. As conclusions of the whole talk, we can say that nanoparticles have several properties that make them unique in their interaction with biological system. They may have biological actions by themselves, like anti-inflammatory properties. Some of them can cross the BBB to deliver therapeutic molecules like drugs or genetic material to the brain. But for those that doesn't do this, it is also possible to decorate the nanoparticles with chemical groups to increase BBB crossing. They can be selectively directed to different cell types, and as a corollary, nanoparticles are a group of chemical compounds with high potential to be used in the therapeutics of different diseases, including neurodegenerative diseases. Thank you very much. Oh, sorry. The most important one is the people that allow me to present this data from my group. So all these people, basically mainly Inmaculada Posadas and Ana Belén García, these are 3 persons, chemists, with which I collaborate regarding this molecule. We collaborate with many more chemists, but these 3 have been involved in this work, like Jean-Pierre Majoral, Don Tomalia, and Sofia Lipovska. Thank you very much.
Yeah. Who's got the first question? Who's got the first question?
Have you tried anything in the typical neurodegenerative disease like Alzheimer's, Parkinson, and so on? Do you have any data, or do you know about any data using nanoparticles with these kind of diseases?
Not in Alzheimer's disease. You mean in whole animals, animal models?
Right.
Yeah. No, we are planning to start shortly something with animal models of Alzheimer's disease. We, as everybody here, run sort of funding, but now we think we can start now.
Any other question? Yeah. Then we carry on.